It's my true pleasure and privilege to present you before the major research findings of my doctoral research program on genome-wide search for genetic diversity, selection signatures, and breed-specific markers in native goat breeds of Kerala. Actually, I was very fortunate to have an advisory committee that was chaired by Radhika ma'am, and the members include Arvindasha sir, Director CACGB, Tirupati sir, Professor and Head University Goat and Sheep Farm, Raji ma'am, Associate Professor, Department of Veterinary Physiology, and Shainu ma'am, Associate Professor, again in Department of Veterinary Biochemistry. So, when we plan our work, our intention was to have the maximum utilization of the high throughput genomic data that we are going to have. So accordingly, we designed this work with four major objectives. The first one was the genome-wide assessment of genetic diversity among native goat breeds of Kerala. And second objective was to do the genome-wide search for selection signatures to identify the genomic areas that are divergently selected into different breeds. Diversity matrix, that is the linkage disequilibrium. And here it is the non-random association of alleles between two loci. And the most important application of linkage disequilibrium is the basis of linkage disequilibrium between the adjacent markers is the basis of accuracy of genome-wide association study and genomic selection. Here also, we can see that the linkage disequilibrium that measured in terms of R square was very low. That is only 0.06. Both in Attapadi as well as Malabari goats, indicating the high diversity within population diversity in both breeds. And from the information of linkage disequilibrium, we calculated the historical effective population size in both herds, and we can see that in Attapadi it is 371 around 17 years back, whereas in Malabari it is 518, indicating that both breeds are not at any immediate risk with respect to their management is concerned. And here the model-based structure analysis also shows the distinct ancestral influence of the two breeds. So, analysis of genetic diversity, the conclusion is that both within and between population diversity is very high, and they have a very low level of autozygosity, low level of LD, indicating the high effective population size, and the distinct structure of the two breeds. Coming to the results, the outline analysis that detects the ancient selection signatures, by the outline analysis, we could see 104 outlier loci at a very high level of significance at genome-wide level. And there are many genes, and some of them are FGFR2 and GLI2 that is important for the mammary gland development. And we could also identify some genes that is related to the coat color differences among the two breeds, and the genes we could also identify some genes that is related to the reproduction, production differences among the two breeds. And coming to the results of the haplog analysis, we could identify 7 important peak regions that are under selection during the recent times, and the maximum peak were observed on chromosome 6 and chromosome 12, and this the area in chromosome 6 is very important in the sense that it was detected in the earlier analysis also. And these were the important genes, very important. We had a lot more genes, the important genes in the haplog analysis that we got.